Okay, so in Chapter 4 we learned about hydrocarbons and the different functional groups that play a role in giving organic compounds specific properties. And so now we're going to build on that and look at some different molecules that are important for living things that have those functional groups as a part of them and look at their function and their roles in cells and in living things. So the overview here is within cells. So in cells we have small organic molecules, and remember organic means it contains carbon, and they're joined together to form larger molecules. And we're going to talk about how that is here in just a second. And so we have small and large organic molecules, and the largest ones, we call them macromolecules. They're large molecules composed of thousands of covalently connected atoms. So they're really big molecules. Macro is the opposite of micro. Micro means small. Macro means big. And so they're very large molecules. And so how those are formed, they're formed from smaller molecules. And so most macromolecules are what we call polymers. So let's look at that word polymer. Poly means many. And so polymers are molecules that are large, that are made out of many smaller molecules. And notice here it says built from monomers. Monomer means one. And so these are smaller molecules, and so monomers build and hooked together to make polymers. So an idea, an analogy with this is that a train. So if you have a train, and here are all the cars in your train. Here's your little train. All right. And so on and so forth. This whole thing, the whole train, is the analogy for your polymer. And then each individual car, each car represents the monomer. So that's the relationship between polymers and monomers. So when we write here, a polymer is a long molecule consisting of many similar building blocks called monomers, right? And there are 4 classes of macromolecules. Only 3 of the 4 classes are considered polymers, are made out of many smaller molecules hooked together. And those 3 classes are carbohydrates, proteins, and nucleic acids. And I'm going to write over here that lipids are macromolecules. So we're going to learn about lipids, and they're really big molecules, but are not polymers, meaning that they don't have repeating units like this, all right? But they're still, sorry, I'm off the screen here, but they're still really, really big molecules, and so therefore can still consider macromolecules. So let's look at how these polymers are both made and broken down. So monomers form large molecules by condensation reactions called dehydration reactions. Sometimes also called dehydration synthesis. So you can see reaction or synthesis. Both of them are referring to the same process. So what do you think of when somebody is dehydrated? What are they missing? What have they lost? Water, right? And so that's what actually happens. So you form larger molecules by condensation reactions called dehydration. So another way we can call them condensation reactions, dehydration reactions, or dehydration synthesis. And so all of that is losing a water molecule to build a larger molecule. So and then the opposite, polymers are disassembled back down to their monomers by the opposite reaction, and this is called hydrolysis. A reaction that is essentially the reverse of dehydration. So this we lose a water, this we add a water. So let's look at this picture down here. All right, so I want to point out, I'm going to divide this up. There's two different things going on here. So we have part A and part B, and so we can easily see that. And so part A, let's look at this. This is your dehydration reaction. It says in the synthesis of a polymer. So we're making a polymer. So here, notice here we're starting out with a short polymer. So each one of these is the individual molecule. Our individual monomer. So there are 3 monomers bonded together to make one polymer. And so what we want to do is we want to make it bigger. So how do we make it bigger? We add another monomer here. And so notice here, it's not showing you, this is just a generic picture, so it's not showing you the chemical structure of these monomers and so on, but it is showing you some atoms hanging off of here that are important in the bonding of the new monomer to make your longer polymer. And that is the H's and the OH's. And so notice here, I'm going to use, let's use a different color here. Let's use a lighter blue for water. And so what happens is when these two come together, this one, the H and the OH come off and then collectively come together and make a molecule of water. So then what happens then is when the H and the OH, these bonds break, this bond and this bond right here, they're no longer bonded to each other anymore, or to the OH and the H, so they're not bonded to anything, so they're looking to bond with something. And so you know what they do? When the H and OH come off, they bond together, and that's this new bond right here between the 3rd and the 4th monomer here, and now we have a longer polymer because it is one more monomer long. And so this is why we call this dehydration reaction, because in the dehydration reaction we lose water molecules. So therefore one comes off. So every time one is bonded to another monomer, you lose a water molecule. And so so all of our carbohydrates, proteins, nucleic acids, and even lipids are built using dehydration reaction. On the flip side, when we break down molecules, so let's say we have a large molecule, this is the opposite. It's called hydrolysis. Let's dissect that word up here. Hydrolysis. All right, so hydro is referring to water. Lysis means to split. So what we are going to do is split or break down this larger molecule by using water, by adding water. So here you have a polymer that's 4 monomers long. And so what we're going to do is we're going to add water. So I'm going to use that same color. Add water. And notice here, when you add the water, the H goes on to one and the OH goes on to the other. So if we look here at this bond that's being split, the hydrolysis, splitting with water, this, the H and the OH, this, whatever atom is in monomer 3 that's making this bond with 4, that atom is now bonded to the H, and this atom is no longer bonded to the molecule 3, it is bonded to the OH, and now we have, in this case, right here, a shorter polymer, and this is a monomer here that is broken off of that now. And so to continue breaking this down, we would need how many more water molecules? We would need a water molecule here and a water molecule here, right? And so every time there's a bond, you would need a water molecule to break it down. And so this is like what happens during digestion. But both of these here processes are what we call this reaction here with this water coming off or the water coming in. We call them enzyme-mediated reactions. What does that mean? Well, it means that enzymes have to help it along. What do enzymes do? Enzymes help speed up chemical reactions. So therefore, this is helped by enzymes. And so enzymes have a big role in that. And so this down here would be like digesting food with the help of enzymes, and this would be like building. So once you digest your food, let's say you digest it into your sugar glucose, and the glucose goes into the cells, and your cells put glucoses together to form storage molecules. And so therefore, you can store molecules this way. And we'll go over examples of this as we go through the carbohydrates, which is what we're going to do next. All right, so let's look at. So the diversity of polymers, each cell has thousands of different kinds of macromolecules. So we have a large variety. Thousands. Macromolecules vary, though, among cells of an organism. So the cells of an organism have different molecules in it because different cells of the organism have different functions. They vary more within a species. So from person to person, let's say, or from, worm to worm, and they vary even more between species. So looking at two different species, like a bird and a crocodile. So an immense variety of polymers can be built from a very small set of monomers, and we'll look at examples of that as we look at each of the types of polymers. So let's look at polymer group number 1, the carbohydrates. So carbohydrates. All right, so I'm going to put here polymer group 1. To kind of give you some organization to the notes and kind of color code and so on. So I'm going to switch colors to write about carbohydrates. So carbohydrates include sugars and the polymers of sugars. All right, so we can have one sugar and chains of sugars. The simplest carbohydrates are called, you've probably heard of this word, monosaccharides. And there's our word, our prefix mono, meaning one, and so that's the single part, and saccharide means sugar. So single sugars. So these are the monomers for carbohydrates. That's the simplest thing that you can have. Carbohydrates are macromolecules, and they are the macromolecules. These are simple sugars, one sugar molecule. The big ones that will have many sugars together are called polysaccharides. Polymers composed of many sugar building blocks. So let's look at the simplest sugars here. So sugars, monosaccharides, have the molecular formula that are usually multiples of CH2O. So when there's not a number here, we're assuming that there's a 1. What does this mean? It means that there are twice as many hydrogens as there are carbons and oxygen. And glucose is one that you've heard of before, hopefully. It's the most common monosaccharide. Do you remember what the formula, the chemical formula for glucose is? It's C6H12O6. All right, so hopefully that rings a bell. And so notice here it follows that rule. It has twice as many hydrogens as carbons and oxygens. All right, and so that's your monosaccharides. And they're classified by location of the carbonyl group and by the number of carbons in the carbon skeleton. So remember we talked about carbon skeletons in that you can have 4 carbons or 8 carbons and so on. And so therefore monosaccharides are classified based upon that. Let's review before you look down here. What does the carbonyl group look like? See if you can remember what that looks like. It's a C with a double bond to an O. Okay? And so that's what you're looking for. And then the C is going to be bonded to the larger molecule as a whole. All right, so let's look at our classification of these guys. So let's just look at how this is organized. It says ketoses and aldoses. Whenever you see ose at the end, all right, ose, so that means it's connected to the larger word. That is referring to a sugar. So even if you've never heard of it before, so notice glucose is a sugar. All right, so ose is a sugar. So we have these categories of sugars, aldoses and ketoses. And what do you think that plays a role in that? Aldose comes from aldehyde, and ketose comes from ketone. All right, so these are aldehydes. These guys are ketones, and that has to do with what? The location of the carbonyl group. So let's review that. So these guys are going to be, have the carbonyl group and are going to be aldehydes, and these also have the carbonyl group, but these are ketones. Now notice here, triose sugars, pentose sugars, hexose sugars. Tri meaning 3, so C3, pent meaning 5, C5, so there's 5 carbons. Hex, C6, that's a typo on your thing. It should be C6 right there, so all right, instead of C5. And so there are 6 sugars. And so these are the looking at the columns. And so let's look at, these are aldoses that are, this one is 3 carbons. So 1, 2, 3, triose. It's an aldehyde. Aldehyde means the carbonyl group. The C with the double bonded O is at the end. Aldehyde, end. So therefore, we see that here, pentose, 5 carbons, aldehyde at the end. Hexose, 6 carbons, carbonyl at the end, all right, and the carbonyl at the end here, all right. So this is glucose and galactose. And so I'm going to talk about in just a minute these guys here. I'm going to hold off on that for a second, the highlighted parts here. And then these are your ketones. Again, this is a triose just like this, but it's a ketone. Why? Because the carbonyl group is in the middle. And so this is 5 carbons in the middle, 6 carbons in the middle. All right, and so let's look at these guys here. The hexose sugars are important sugars. Here's glucose here. Glucose is C6H12O10. This is the same for all of these. So glucose, galactose, and fructose all have the same formula, but yet their structures are different. So how are their structures different? Well, this one, the carbonyl group's in the different location than these two. Even if the rest of it, you know, down here is the same, that different location of the carbonyl group makes it an isomer. These two are isomers as well, so the carbonyl group is in the same location, but notice that's what this highlight part is. These are the 2 parts that are different between them, and therefore make them isomers, all right, of each other. And so that is your hexose sugars. Now, this is we're going to talk about this. This is a linear form, and this is a linear form of the pentose sugar and so on. Most sugars in living things are found in water, and we're going to see in just a minute that it actually forms a ring when put into an aqueous solution, into water. And so glucose, you probably learned in biology, is a ring-shaped sugar, and that is the case when it's in water. Okay, so let's look at the next page here. So what's the purpose of the monosaccharide? You use glucose for energy. So it's the major fuel or energy source for cells, and as a raw material for building molecules. And so we're going to talk about some ways that some molecules that are examples of building. So as I talked about just a minute ago, though often drawn as a linear skeleton in aqueous solutions, they form rings. And so this picture here is showing you that formation from linear to ring shape. Now with this is figure 5.4 in your textbook. You don't need to know and be able to explain this reaction. This is just showing you how glucose goes from linear to the ring structure here. And so I'll do just a quick little thing here. They number the carbon atoms, in the sugar, and so 1 through 6. And so what happens is if you imagine if I were to pull this up here, so now if we look over here, this part right here, This is the 6th. All right, so that's CH2OH, CH2OH. All right, so that's up here. So this atom is right here. All right, and so I'm going to put this here in blue. All right, and this part right here, that's this in blue right here. Okay, and this right here in red here, and that's this part right here. So you pull this up along here so that this is hanging off. This is this carbon that has the OH, and this is the carbonyl group. And so what happens is that, let me get a different color here, is that this H comes over here to this, all right, and breaks this so it's not a double bond, and then reforms a bond right here. And so this is the new bond right here. And so this was the O of the original OH, and down over here is the H that was originally up here, and then here is your CO and H. Now, I just did that real quickly for those people who are interested and understand chemistry very well and just kind of want to know. But for the rest of you, you just need to know that it shifts, can shift from linear to ring-shaped, and this is what it looks like in the ring-shaped. And so we find this in the ring shape in aqueous solutions. So up here in the aqueous solution. So from now on, from here on out in our textbook, since we're talking about biology and the living things, you will see the glucose in a ring shaped. And so it's going to look like this. And then one other point I want to make is they abbreviate that ring structure. So from now on in our textbooks, oftentimes they won't have the carbons in there. They'll just have the points. And you are supposed to know that each of the points represents a carbon atom without them having it in there. Okay, and so they have them numbered here just like that one. So that's the abbreviated ring structure, and so that's what we're going to see from now on. So that is your monosaccharide for fuel and energy and used to build larger carbohydrates. So now let's look at what happens when we take two rings, two sugars, monosaccharide, and hook them together. Now we don't have a monosaccharide anymore. We have two sugars hooked together, and therefore that is a disaccharide. Disaccharide is formed when a dehydration reaction joins two monosaccharides. So now we go back to that original picture. You know, we were talking about dehydration building a larger molecule. Well, in this case here, we're putting two monosaccharides together. So two of these here together, how do we do that? We take an OH off of one and H off of another, and that is dehydration, or dehydration reaction, or dehydration synthesis, or condensation. And so, so they call this bond between the two a glycosidic linkage. So let's look at this picture here. And again, this is 5.5 in your textbook. So this shows you making the disaccharide maltose. All right, so here's maltose. So here are your two monosaccharides. Maltose is made from glucose and another glucose hooked together. And so, again, I do this color here. So here are your two groups. This is an OH group and an OH group. And so what happens is these guys here in this OH, you lose an OH and an H in dehydration reaction. And so now this carbon that was once here, this bond, and this oxygen is no longer bonded right here. And so therefore now they are going to bond. So this oxygen right here is this oxygen here, and the new bond is right here. Okay, and so therefore now this is a disaccharide called maltose, made out of the joining of two monosaccharides called glucose. And this is, it says a 1,4 glycosidic linkage. What they do is they number the carbon atoms. It's between the number 1 carbon atom of this glucose and the number 4 carbon atom of this glucose. And so then we look at sucrose. Sucrose is made from two different monosaccharides. It still has a glucose, but it's also fructose. So glucose and fructose. And again, you have an OH and an H comes off. So this is also dehydration. And so this oxygen will now bond to this carbon. And so this oxygen right here is right here, and this is the new bond. And so so that's what we have here. Fructose, notice fructose is one of our ketones here. So it's still a 6-carbon sugar. And so now we have our table sugar. Okay, and so they lost 1 water molecule in making the table sugar. Okay, and there's actually one more disaccharide that we'll talk about sometimes in this course, and that is lactose. And lactose is made from glucose plus galactose. We'll give you lactose. And so this is a monosaccharide, disaccharide. Okay? And so they're all made the same way through dehydration reaction. And our last group of carbohydrates are your polysaccharides. So we've had monosaccharides, disaccharides, and polysaccharides. And we kind of jump hugely in size right now. So we go from 2 simple sugars hooked together, that's your disaccharide, and polysaccharide is many, many, many. So it's way bigger. So polysaccharides, the polymers of sugar, have both storage and structural roles. So the structure and function of a polysaccharide are determined by its sugar monomers. So what it does and the formation it takes depends upon what sugars are hooked together when making it. And then also the positions of where their glycosidic linkages are. So that's the bond between the 2 monosaccharides. So let's look at storage polysaccharides first. So a huge one is starch. We've all heard of starch. Starch is a storage polysaccharide of plants, and it's made out of entirely of glucose molecules. So it's a big chain of glucose molecules is basically what it is. And so plants store surplus starch as granules within chloroplasts and other organelles called plastids. Chloroplasts are a type of plastid. Chloroplast is where photosynthesis occurs, but they can store it in other places as well. And so this is actually a picture inside of a plant of this whole picture right here. This is the whole chloroplast right here inside the plant, and these are the pockets of where they store starch. So remember, in the chloroplast, that's where photosynthesis occurs, and what does photosynthesis do? That's how we get glucose on the earth. So they make glucose, and so when they make glucose, they can use the glucose, some of it right away, and then some of it they store it. And how do they store it? They hook them all together into a molecule called starch. So when we look at this, starches, these are 2 different kinds of starch. You don't need to know the differences between them. So both of these are types of starch. What you do need to know is that all types of starch are chains of glucose molecules. And so each one of these little rings right here, each one of these rings is a glucose molecule. And so it's a chain of glucose molecules. And so the plant makes, so let's write here, the plant makes glucose through photosynthesis. And can do one of the following. It can use the glucose right away for energy. Or B, they can store it in chains called starch for later use. So what does that mean for later use? So what they'll do is if they need energy later, they will break down the starch, and when you break down starch, you break down the bonds and you end up with individual glucose molecules. So therefore they just store it for a while, and then, oh, we need some energy, and so therefore we start breaking it down, and instead of one starch molecule, you can have thousands of glucose molecules, and you can use those for energy. And so that's starch, and that's only found in plants. So we eat plants and eat their storage. So for instance, potatoes are a huge storage of starch for the potato plant, and we end up eating that, and so therefore we're eating chains of glucose, and then we can in turn eat it and break down the starch into glucose, and then use that glucose for energy as well. Or sometimes we eat too much glucose. And when we eat too much glucose, our body stores the glucose for later use as well, but we don't store it as a molecule of starch. What we store it is as a molecule of glycogen. So glycogen is a storage polysaccharide in animals. And so I'm going to put here like us as humans. And so humans and other vertebrates store glycogen mainly in our liver, in our muscle cells. Muscle cells are important because they use a lot of energy. So we have the storage right there in our muscle cells. The rest of the body relies on the glycogen stored in the liver. So when you eat too much and you have too much glucose and you don't need it for energy right away, then what happens is all of that glucose, when you break down the starch in your digestive system is going to break the starch down into glucose molecules. The glucose is going to go into your bloodstream and carry to all the cells of your body. But guess what? If you don't need the glucose for energy right now in the cells, then that glucose is going to leave your bloodstream and go to your liver, and your liver is going to hook them all together to form glycogen and store that glucose until you need it again later. So if we look at glycogen, it's more highly branched than starch. But like starch, it's still a chain. So you can see all it's still a chain of glucose. You can see all the branches here and so on. And so we would store this in let's say our liver. And then what happens is then let's say you go run a 5K or something like that and need a burst of energy. What your liver will do is, as enzymes that will break these bonds and break your glycogen into, instead of one large glycogen molecule, it'll break down into thousands of glucose. Those glucoses enter your bloodstream and you can use them for energy. And so that is glycogen. And so those are your two storage polysaccharides. So then in plants and in animals. So then there are two other types of polysaccharides, and these are examples of your structural polysaccharides. So these are polysaccharides that are not broken down and used for energy at all. They are used to help build parts of your cells or parts of your body. And so in plants, that is called cellulose. It's a major component of the tough wall of plant cells, major component of it. So in this case here, when we look at the cell wall, plants have the cell wall, and then they have the cell membrane underneath it, like this. All right, so we're talking about the tough cell wall. And it has, so the plants need this tough cell wall. Animal cells don't have one. They need this tough cell wall because to give the plant cell structure and the plant structure, because guess what? Most plants do not have skeletons like we do, and so therefore they rely in part on the cell wall for structure for the plant. So like starch, cellulose is also a polymer of glucose, so it's also a chain of glucose. So every polysaccharide we've talked about so far is a chain of glucose, but the glycosidic linkages differ. So how they're hooked together differ. And the difference is based on two ring forms for glucose. They call it the alpha and the beta ring form. So let's look at this. So what's the difference between an alpha and a beta glucose? So here's in the middle here. This is the linear part of the structure of glucose. So this is the alpha and this is the beta glucose. And so basically if we look at the difference, so remember each point here is a carbon atom and you have H and OH and H and OH and all of that's the same. The only thing that's different is this part right here where this the carbon atom number 1, the OH down here is coming off to the bottom, and this one is coming off to the top. All right, and so let's look at starch versus cellulose. These are both found in plants, right? So starch and cellulose both found in plants. So both of these are found in plants. This one's the starch is the plant storage polysaccharide. This is cellulose is the plant structural polysaccharide, right? And so notice here starch. They're both chains of glucose. Starch is made out of alpha glucose molecules and cellulose is made out of beta glucose molecules. And this is a big deal and changes the structure of it. And let's talk about why. So alpha glucoses, this is one alpha with another and so on and so forth. And so all of these are the glycosidic linkages where the sugars are hooked together. And so I want to talk about how that happens. So when you hook the two sugars together, two glucoses here together, what happens? What's it called? It's dehydration, and you lose a molecule of water, right? And so they're all made of a chain of alpha glucose molecules. So let's draw the alpha glucose molecules over here. So here, starch, I'm going to put it up here so I have enough room. I'm just going to draw here's the ring of the glucose, so that's this part here, and I'm just going to draw the parts, the two end parts, because that's where they hook together. So that's what matters: the H and OH and OH and H. So you see that here? All right. So that's one alpha glucose, and then it's a chain of alpha. So I'm going to go ahead and draw the second alpha here, glucose, and so it's exactly the same, OH and H. So these are two alpha glucose molecules. So let me switch colors here. So can you see that the hydroxyl groups line up here? And so remember that you lose an OH off of one and H off of another. That's your dehydration. You lose a water molecule, and now this O binds to this carbon here, and now you have them hooked together. All right, and so therefore that's two alpha glucoses hooked together. And then you could have do it for the next one. So you could have a third. Let's just do it one more time. So here we have another alpha, and then here H, OH, H, OH, and again OH and H come off, molecule of water. Oxygen binds to that carbon, and so now you have three, and so on and so forth, and you can continue to do that, and that's how starch is built. All right, so when the plant makes the glucose and wants to store it for later use, this is what it does. And so this also is helped with enzymes. So now let's see the difference between that and glucose. So the plant also takes some of the glucose and makes cellulose, and this is for cell walls, and it has a different structure, and let's look at why that is, and that's because it's made out of these beta glucose molecules. So now I'm going to draw the beta glucose molecules. So maybe I should do it in a different color here. So the beta glucose molecules. Now what is the difference between the beta glucose molecule? And so when we do this, beta, let's look at this, has H and OH. That part is the same as the alpha, but look at here, what switches is the OH and the H. So this is a beta molecule of glucose. So then we're going to bind it to another beta because it's a chain of beta molecules. And so in this case here, so we have the OH and the H, H, OH. So this is another beta. Do you see now when these guys try to bond, remember an OH and an H have to leave. So if you notice here that these guys here are not lined up. It's the H of this group and the OH of this group. They're not lined up here. So what has to happen in order for them to line up is, can you see that you have to flip this one upside down? So let's look at this that, I'll use orange, I guess. So this one needs to flip upside down in order to make, so if we actually picked this up and flipped it, do you see where the OHs would line up? So if we did that, let's make this so now the OH would be here, and then the H would be down here, and this one, if we the OH would be down here, and there would be just an H here. So that's what that would look like now. All right, so now you have the OHs lined up, and so now what can happen is OH can leave, H can leave to form a molecule of water, and then this O will bind to here. And so if we look here at this and over here, this O is this O right here, and this bond right here is that new bond that's formed. All right, so that's the new bond that's formed there. And so notice here that it goes up. So then let's look at the problem that happens now when we try to hook another beta glucose molecule. So again, I'm going to draw the ring, and the beta has the OH and the H and OH here. So now that this is flipped, now notice that these guys here line up, and we see we can lose an OH and an H in a molecule of water. This O will now bind to this carbon here. And so now notice the bond is down at the bottom here because that's where the OHs were. And so then you see here, do you see it here, down here below? So this bond right here is the new bond. So see how the O goes up and the O goes down, and then we can continue to do that. So you can see that if I do another beta, all right, I'm going to have the same problem that I did originally here, and then we'll have to flip it upside down and so on and so forth. And so you get these alternating kind of bonds here. And therefore that actually affects the structure of the molecule, which actually makes it a stronger molecule. We'll see that in just a second, and that's why it's used for structure. And it's just because of the monomers that are used and the way that they have to flip and so on. So polymers with alpha glucoses are, and the shape of those are, let me get my carbohydrate color here, are helical. So that's starch, all right? So starch up here, these guys would be helical. And if you remember that when we showed the picture over here of starch, it's in a helix, kind of like a telephone cord. And polymers with the beta glucoses are straight. So it changes the structure because of those different monomers. So return the page. So in these straight structures like cellulose, hydrogen atoms on one strand combined with hydroxide groups on other strands. So they bond together, and looking at this here, it says parallel glucose molecules held together this way are grouped into microfibrils, which form strong building materials for plants. And so that's what we're seeing here. So you have, this is one, so it says here cellulose molecules. So this is one chain of beta glucose molecules, another chain, and we see that alternating up, down, up, down, up, down of the oxygen there. So these are different chains. These guys bond, so they're held together. It says here the hydrogens on one strand can bond with OH groups on another strand. So that's what these bonds are right here. All right, so that's what these dots here, OH, All right, they're showing you the bonding between this. So these guys kind of bond together and come together. And so you can see that they all come together to form a microfibril. And so then you have multiple microfibrils that crisscross, kind of like threads in a shirt. And they go in different directions. Why? Because that gives it strength and makes it more durable. And so that is what the cell wall is made of. And so it's all of these cellulose molecules bonded together that come together in a microfibril, then they crisscross and so on and weave around each other, and therefore add strength and stability to the cell wall, and therefore helps give the plant structure. So one thing about cellulose, so cellulose is the structural material for polysaccharide for plants, is that enzymes that digest starch by hydrolyzing, so we can eat starch. We eat a plant, we eat a potato, we're eating starch. We have enzymes in our digestive system that digest starch. They break the bonds and break it into glucose molecules. So enzymes that digest starch by hydrolyzing alpha linkages can't hydrolyze beta linkages. What does this mean? That we can't digest cellulose. So even though cellulose is also a chain of glucose molecules, those beta linkages and our enzymes do not work to do that. So we don't digest cellulose. So cellulose is often in that clump of food that's called fiber. So cellulose in human food passes through the digestive tract as insoluble fiber. Insoluble means that we can't digest it, so it goes right through our body and leaves in our solid waste. But fiber is really good for us. And so fiber actually helps add bulk to our solid waste and helps it move through our digestive system in a timely manner, so it doesn't sit in our digestive system very long. And so fiber is a good thing to have, so keeps things moving through. So some microbes, though, microbes are small organisms, like microscopic bacteria would be a microbe. Some microbes use enzymes to digest cellulose. So we don't have enzymes to digest cellulose, but other organisms do, and so some bacteria are examples of that. And many herbivores eat plants, so from cows to termites, have what are called symbiotic relationships with these microbes, meaning that they help one another. So the cows will have bacteria in their gut to help them break down the fiber, and then they provide a nice warm spot for the microbes to live, and so they're both benefited from that relationship, and that's called a symbiotic relationship. And then the very last carbohydrate is called chitin. Chitin. Chitin is another structural polysaccharide, and this is found in the exoskeleton of arthropods. Exoskeleton means it's the skeleton on the outside of the body. And so, chitin provides structural support for the cell walls of many fungi. That's where we find it. And also is so strong that it has been also used for surgical thread. All right, and so it is extremely strong here. And so chitin is this is showing you the exoskeleton, but chitin has the formula, or it's a ring structure, and it's unique in that it has nitrogen as an element. It's the only one that we've seen so far that has nitrogen as an element. Most carbohydrates have just carbon, hydrogen, and oxygen. So C, H, and O, and then chitin is an exception in these the fungi or the exoskeletons that have nitrogen. And so that is also a type of structural polysaccharide, and that is carbohydrates in a nutshell.